So thank you everybody for joining us in what I think is now the 11th or 12th Plant Phenome Journal monthly webinar series. So my name's Seth Murray, and I'm an editor and was the one who handled the speaker's article. And so I'll be moderating the discussion and helping with the webinar. So Dr. Larry Purcell is a distinguished professor and Alzheimer Chair for Soybean Research at the University of Arkansas. Dr. Avi Kaler is a postdoctoral scholar who did a lot of the work with Dr. Purcell. And then Sumandeep Bazzer is a graduate student who is also online. So they're going to be talking about a recently published paper that they submitted to the Plant Phenome Journal, Carbon Isotope Ratio as a High Throughput Measure of Water Use Efficiency in Soybean. So you can watch the past webinars, the ones that were successfully recorded at least, on our YouTube channel. So you can just search the Plant Phenome Journal. And you can also rewatch, hopefully, any of the things you heard here today. And you can also learn a little bit more about cutting-edge work going on in plant phenomics. So after Dr. Purcell gives his seminar, I will open it up for questions, both orally and in the chat box. If you do have any questions throughout the webinar, please go ahead and just type it in the chat box or email me. I'll be monitoring those throughout the day. So without any further ado, Dr. Purcell.
Thank you, Dr. Murray. I appreciate the invitation, and I appreciate the opportunity to share with you and others joining us the research that we've conducted looking at carbon isotope ratio. Just as a way of getting started, I also want to recognize the United Soybean Board for generously funding a project that allowed this research to come about. So just as a way of starting out, well, let's see. There we go. Just as a way of starting out, the manuscript that we published in Plant Phenome Journal also had as co-authors, in addition to Dr. Kaler and Sumandeep Bazzer, Alvaro Sanz Saez, Jeffrey D. Ray, Felix Fritchie as co-authors on that, and I want to recognize their contributions to the paper itself. So the reason that we're interested in carbon isotope ratio is that it can be used as a surrogate measure of water use efficiency. And water use efficiency is certainly something that is of great interest to people interested in stress responses in crops. But it's very difficult to measure accurately in the field. And having some method of establishing differences in water use efficiency in crops and among genotypes within a species is certainly something that we were very interested in doing. The objective of our research was really quite simple. We were interested in comparing the C13 ratio among tissues collected at flowering, as has traditionally been done, with grain harvested at maturity. And the rationale for this was that in just about any agronomic type of evaluation, the grain is going to be harvested normally, and it would being able to use the grain as a source of the C13 ratio would be much greater. It would improve the utility of looking at C13 ratio rather than having to take a separate sample at some point during the crop growth. So that's really the objective and the rationale for this research. So since we're really talking about, when we talk about C13 ratio, we're really looking at water use efficiency. And water use efficiency comes down basically to the uptake of carbon dioxide during photosynthesis at the expense of water vapor leaving the plant. And so it's this dynamic ratio of carbon uptake and water loss through transpiration that drives water use efficiency. And we can express water use efficiency as just the ratio of photosynthesis and transpiration. And on a longer term basis, we can think about water use efficiency as the accumulation of biomass and the loss of water during the period when biomass was accumulated. So, you know, just generally thinking that's what we want to think about with regards to water use efficiency. I want to go back in the literature just a little bit and talk about water use efficiency and some key papers that came out over the years looking at water use efficiency. And the first one came out in the early 1900s, Briggs and Schatz in 1914. I want to talk about it This was followed up in the 1958 by a very thorough review of the literature that DeWitt did, and his conclusions from that. And then following that, a review article that Tanner and Sinclair published in 1983 that really took a mechanistic look at the components of water use efficiency and what those actually mean physiologically. So first, let's look at Briggs and Shantz. They had an article published in 1913, actually two in 1913, I believe, 1914, and then Shantz and Piecemeal in 1927. And so in this graph, we're looking at biomass accumulation versus transpiration in several different environments. This is all in wheat and all with the same cultivar at wheat, but just grown at different times and in different years. And so what we see here is that in each case, there's a linear relationship between biomass accumulation and transpiration. I want to talk a little bit about Briggs and Shantz and what they did, because it was remarkable work and incredibly extensive. They looked at over 100 species of plants and potted studies, and what they called potted studies, but these were actually tubs that had 115 kilograms of soil per tub. These were sealed at the soil surface with a wax tallow mixture to prevent evaporation from the soil surface, so they were actually just looking at transpiration. And at the peak, they were weighing about 300 pots 3 times a week using this rather elaborate system. Within an environment, like I said, they found a linear relationship between dry matter and transpiration. They saw large differences in transpiration among environments, as we can see here. And they noted that differences in transpiration were proportional to pan evaporation. They also noted that there was a greater slope for tropical grasses than wheat. So the slope of this line here would actually represent water use efficiency. And what they were seeing was what we now know is differences between C3 and C4 species. DeWitt took a lot of the research that Briggs and Shantz and others before him did and reanalyzed it. And this is a plot of data from DeWitt that I've replotted here. We've got biomass on the y-axis, transpiration normalized by pan evaporation on the x-axis. And over many diverse environments, we see a very, very linear relationship for sorghum here at the top line and wheat at the bottom line. So again, it's a way of normalizing the data to account for differences in the environment. So dividing the transpiration by the pan evaporation resulted in a linear relationship among these diverse environments. And DeWitt's analysis said that biomass was equal to this coefficient M multiplied by the transpiration, and that was divided by the pan evaporation. And rearranging this, water use efficiency is equal to M divided by the pan evaporation. So the values of M, this coefficient that DeWitt came up with, were species dependent and were stable across different environments. Tanner and Sinclair in 1983 published this seminal work that really took off where DeWitt's analysis left off. This is the equation that we just looked at from DeWitt, where water use efficiency is equal to M divided by pan. And they asked the question, what is the mechanistic meaning of this M value? What does it mean? And so they came up with a new expression. Water use efficiency is equal to this value K divided by the vapor pressure deficit. So they looked at K and broke this K value down into several different components, and I want to run through those briefly. The A coefficient here is the molecular weight ratio of going from carbohydrate to CO2. You know, we're taking carbon dioxide up in photosynthesis and initially converting that to carbohydrate. The B coefficient here is the conversion of carbohydrate to biomass. And this is important when you think about different species and the dry matter and what it's composed of. For example, if a crop is composed mostly of carbohydrate, it is much less expensive to produce energetically than if that plant dry matter is composed of oil and high concentration of protein. The C coefficient here is equal to 1 minus the CO2 concentration inside the leaf divided by the CO2 concentration in the surrounding atmosphere. And this is multiplied by the CO2 concentration in the atmosphere. So this actually, the C times the C atmosphere actually reduces to the ratio of CO2 concentration inside the leaf divided by the CO2 concentration outside the leaf. The LD refers to the leaf area in direct beam radiation, and the LT is the total leaf area index. So I want to go, and of course the vapor pressure deficit that we've already mentioned is the vapor pressure deficit that actually occurs during the period when transpiration is occurring. So I want to look at these two highlighted components, the vapor pressure deficit and the CO2 ratio from inside the leaf to outside the leaf. Both of these are very, very important in determining the water use efficiency, and they both have are related back to carbon isotope ratio. So what is the relationship between water use efficiency and carbon 13? Well, when we look at this, the original derivation of carbon 13 in plant tissues was derived by Farquhar and Richards, and they expressed it as carbon isotope discrimination, which is a little bit different from what we've done, as I'll explain. But there's two components in here that relate back to that are important. The 4.4 here is related to how well the C13 CO2 molecule is slowed down in diffusion compared to the CO2 C12 molecule. So that's just a constant in here. But we'll notice here we've got the C13 inside the leaf versus the C13 exterior in the atmosphere. So it's this ratio right here that corresponds directly back to water use efficiency as Tanner and Sinclair proposed. And how are C13 data expressed? I mentioned that Farquhar and Richards used this carbon isotope discrimination expression that's shown here. It is related to the ratio of C13O2 in the air to C12 in the air divided by the ratio of C13 in the plant to C12 in the plant. So that's how they expressed the C13 data. And this is inversely proportional to water use efficiency. We've taken a little bit different expression, as have many others, and we distinguish this from carbon isotope discrimination by using a small delta. And in this case, we're looking at the C13O2 ratio or the C12 ratio in plant tissue, C13 to C12 in plant tissue, divided by the C13 to C12 ratio in a standard. And in this case, the standard's PD belemnite, just a standard that's recognized worldwide. And this is directly proportional to water use efficiency. So that's what we'll be talking about primarily in the work that I'll be presenting. This is an example of looking at the relationship between water use efficiency on the y-axis versus carbon isotope discrimination on the x-axis. This is work that was done in wheat. So we see a negative relationship here where a high water use efficiency has a low value of carbon 13 discrimination. We can also want to look at this vapor pressure deficit and how it's related to water use efficiency in a bit more detail. And We can think about vapor pressure deficit as being tightly linked with temperature. And I've got temperature on the y-axis here versus the time of day. And we see at sunrise, as temperature begins to increase, we have, a nice bell-shaped curve here. As the sun begins to set, of course, temperature begins to drop. And vapor pressure deficit is on the right-hand y-axis here. And we see that it tracks right along with the temperature value. And so looking at this equation at the top, when we think about vapor pressure deficit, as vapor pressure deficit begins to increase, we would expect water use efficiency to decrease. And we can express, in this graph, I'm looking at water use efficiency versus the time of day, and we see that this water use efficiency decreases as vapor pressure deficit reaches its peak. Now, what we can think about is if we have a plant that will close its stomates at some threshold value of vapor pressure deficit, say at about 2 kilopascals, what we would expect then is that transpiration would no longer begin to increase, and water use efficiency would basically reach a flat plateau. And that's one of the mechanisms that we believe is happening in a lot of plants. And this is some research out of Tom Sinclair's lab from 2007, where he's looking at transpiration in soybean and A5959 versus vapor pressure deficit here on the x-axis. And what we see is a linear response of transpiration in response to increasing vapor pressure deficit, which is what we would expect. And a different genotype, in a session PI 416937, we see that transpiration increases up to a threshold of around 2 kilopascals, and then it plateaus and no longer increase in transpiration with increase in vapor pressure deficit. And this would, of course, improve the water use efficiency of PI 416937 under high vapor pressure deficit conditions compared to ASGRO 5959. This is an example of differences that we observe in the field, where we've got a genotype here that in the foreground that is unresponsive to vapor pressure deficit. It continues to lose water, it continues to transpire when vapor pressure deficit is high, and it will quickly exhaust the soil water supply. In contrast, there's in the background immediately behind it is a genotype that is able to restrict transpiration at high vapor pressure deficit, conserve soil moisture, and it would improve water use efficiency when vapor pressure deficit is high relative to the unresponsive genotype. Okay, I want to turn now, I've spent the previous few minutes talking about just background information. This is still background information, but I want to tell you how we selected genotypes that we used in our evaluation to look at the fractionation of C13 in different plant tissues. So we started out with a rather large panel of 373 maturity group 4 soybean accessions. And these were sampled at R2 at full bloom at 2 locations for 2 years. These were analyzed for C13 ratio, and we used genome-wide association mapping to identify 54 SNPs that were associated with the C13 ratio. This is looking at the different chromosomes of soybean from chromosome 1 to 20 on the y-axis versus the chromosomal position on the x-axis. And the red dots identify those significant SNPs that we found associated with carbon isotope ratio. 54 SNPs in all. The black bars here represent the genotypes that we had been identified, the SNPs, the QTLs identified previously that were associated with water use efficiency. And we find there were nine loci that were common between SNPs that we identified and previously identified QTLs that, basically confirmed the presence of water use efficiency with carbon 13 ratio. And this graph, we're still looking at the 373 genotypes from the initial screen. And we're looking at a frequency distribution of the genomic estimated breeding values for C13 ratio. And we selected 20 genotypes from this panel of 373 genotypes across this wide distribution of genotypes in our original panel. So at the low end, the very negative genomic estimated breeding value for C13 ratio, we selected 3 genotypes from this bin, 3 from this genotype, this bin, all the way across. And at the upper end, where we would expect water use efficiency to be high, the least negative values, we selected 6 genotypes out of this bin and 2 genotypes out of this bin. Our purpose here was to get a wide distribution of genotypes with different C13 ratios. So now, in the current study that we published in the Plant Phenome Journal, we selected 20 maturity group 4 accessions. We looked at these at 3 locations in Fayetteville, Arkansas, Pine Tree, Arkansas, and near Columbia, Missouri. We had at each location 2 replications arranged in the randomized complete block design. These were grown under rain-fed conditions, and the plants were sampled at bloom at R2, and then the grain was harvested at maturity for evaluation. So a little bit on how we sampled the plants. From each plot, we selected 5 individual plants. From one plant, we took the center leaflet off of every nodal position, every main stem nodal position. For 3 plants, we chose 3 plants, and we harvested the entire whole shoot, beginning at the soil surface. We cut the plants at the soil surface and selected the entire plant. From one plant, we chose the uppermost fully expanded center leaflet. And then for the mature seed, we just used a combine grain sample that we subsampled for analysis. This is looking at the analysis of variance, the sums of squares that, in this case, from the center leaflet. In our analysis of variance, we found that 66% of the sums of squares were explained by the genotype effect, 11% of the sums of squares by the environment effect, and the G by E explained 20%. This small little wedge up here at the top with the orange color was due to the rep. The stars indicate significance, and the NS, of course, signifies non-significance. So that's the center leaflet. For the whole shoot, we found genotype explained 69% sums of squares, environment 24%, G by E 6%, the upper trifoliate 57% explained by the genotype, environment 22% explained, and for the G by E 21%. And for the mature seed, genotype explained 84% of the sums of squares, environment 3%, and G by E 14%. So for all tissues, the G by E was not significant. The environmental effect was least for the seed. The genotype effect was greatest for the seed. And we also conducted an ANOVA that included the tissue, excuse me, included the tissue in the model, and found that the genotype by environment by tissue was non-significant. And this indicated that genotype ranking for the different tissues among environments was relatively consistent. And looking at the repeatability of the measure, I'm looking at the center leaflet at this point. The open bars are for Pine Tree, the black bars for Columbia, Missouri, the red bars for Fayetteville, Arkansas, and the blue bars is averaged all across all environments. We see that repeatability was ranged from around 28% to about 85% for the center leaflet. For the whole shoot, we see consistently high repeatability from around 0.75% up to around 90%. For the upper trifoliate, a little bit lower than for the whole shoot. We're looking at around 70% up to around 78%. And for the mature seed, again, high repeatability for all locations. So repeatability was high across really all the different tissues that we analyzed, but especially so for the mature seed and the whole shoot. And lastly, I just want to look at the relationship between the mature seed C13 ratio and the other tissues. For looking at mature seed versus the upper mature trifoliate, we see a correlation of 0.78. And for mature seed and center leaflets, the correlation of 0.82. Oh, and I should mention that these values are when averaged across all environments. And then mature seed versus the whole shoot, again, a correlation of around 0.69. The correlations that we saw for all the different tissues in all environments was significant and relatively high. So in summary and just kind of final thoughts here, the genotype by environment interaction was non-significant for all the tissues that we looked at. The environmental effect on C13 ratio was least for seed. The genotype effect on C13 ratio was greatest for seed. The repeatability for all tissues was high, especially for the whole shoots and seed. And for genotypes of similar maturity, which we had in this experiment, the C13 ratio of seed provides a similar inference, as does the C13 ratio from vegetative tissues. I think that's an important point to note here, that all of the different genotypes that we were evaluating were of similar maturity, and had we included vastly different maturities, we may have run into differences in the C13 relationship in the seed, especially because they would have been exposed to different environments throughout the season as the duration of the seed fill period changed. Okay, Seth, I'm going to go back to you, and I'll be glad to answer any questions that come up from the audience.
Great. Well, thank you for your. Can you hear me?
Yeah, I can hear you.
Okay, great. I'm getting a little echo from something. I might mute you while I'm talking here for a second. So thank you for a great seminar, Dr. Purcell. Now, typically what we like to do here is to take questions from everybody. So there's a couple different ways to do that. You can either write questions in the chat section down at the bottom. You can address them to myself. I might have a little more free time to read them than Dr. Purcell, but either one of us. And then I'll also open up the microphone, and you can try and verbally ask your question. So while you're doing that, I'm going to put in a brief plug. I think this was a very nice example of the type of work we like to highlight in the Plant Phenome Journal. Really good interface between traditional sort of plant physiology along with a rapid phenotyping screen. And so as you're thinking about your questions, I'm going to ask a quick one and take the advantage here and say, when you guys published this, one of the questions I think I had for you was, what is the cost, and what is the cost of scaling this technology? And I think you said you outsourced it, but what do you think the future looks like for using this as a rapid phenotypic screen in mapping populations or breeding populations, and the cost therefore?
For each sample, it costs, if I recall correctly, $7.50 per sample, and so it is a bit costly. And I have to say that the preparation of the plant material requires quite a bit of labor as well. The plant material has to be ground to a fine powder. They're only analyzing 2 to 3 milligrams, and the 2 to 3 milligrams has to be representative. And so to get a representative sample of, a kilogram of seed, you have to grind quite a bit and homogenize it to a point where it's basically flour, and that requires quite a bit of effort. And then weighing it out in 2 to 3 milligrams into a small tin capsule is as much of a skill as it is an art, and requires quite a bit of labor. So there is quite a bit of requires quite a bit of effort, and it's not cheap to do. I think that there are possibilities of looking at. There are some analytical methods of looking at C13 using different types of mass spec that I understand are not far away from being available on a wider scale. But I am not very conversant on what those may be and what the future may hold for those.
But I mean, the alternative for what it would have cost you had you actually wanted. I mean, one of the things I think you're showing nicely is that there's some environmental stability there. So maybe with this technology, you don't need to screen quite as many environments. You don't have a big G/E effect. So the alternative to actually running these plots, the cost differential there, what do you think that range is?
I'm not sure I really understand your question, Seth. You're exactly right that G/E wasn't large, and so I think that does cut down on the number of environments in which you might need to run the experiment. Quite frankly, I don't think there's any way possible that these type of data could be collected on trying to measure water use efficiency directly. It's just not possible. So we're excited about it. We know that there's water use efficiency by itself is probably not going to be the answer to improving drought tolerance, but we think that that's an important piece of the puzzle.
Sounds good. I've not seen any questions come up on my chat box. Have you, Larry?
No, I have not.
Okay. So if anybody has any questions they'd like to ask verbally, please go ahead and unmute yourself. There's a little red microphone on the side. You can just click that off and ask any questions you might have. Anyone? Well, in the intro here, because I do find it takes a little while before people really start getting some questions in, would either of the other co-authors like to comment on any of the work that they conducted? Wow, Larry. I'm sorry. It must be after the crop science meeting. Everybody's just beat about some questions. Go ahead.
Whoops. Seth, Avi Abgender is online. He's done a great deal of work looking at other traits, and he may want to comment on some of the other traits that we're also looking in this same panel. Avi, are you there?
He's muted right now.
No. Well, let me fill in just a little bit on some of the other traits that we're looking at in the same panel. Avi says he doesn't have a microphone. Okay. So we're also, I showed differences among genotypes and how quickly they wilt in the field. We've also looked very closely at this in this panel and have mapped wilt, how quickly plants wilt. Using the same panel, we have evaluated canopy temperature from an aerial platform. And it's very interesting because we see these relationships. We see that loci that are closely associated with slow canopy wilting are also associated with cool temperatures during a drought period, which is exactly what you would expect. But in some cases, we get unique loci that for either the wilting trait or the canopy temperature trait that tell us a little bit, or hypothetically a little bit, about what is actually determining this trait of interest. We've also, in addition to C13, canopy temperature, canopy wilting, looked at the fraction of nitrogen derived from the atmosphere using N15 analysis and natural abundance. And in that case, it makes a very nice tie, because when you do the C13 analysis for the Isotope lab, you also get N15 analysis, I think, for $1 more. So you can combine those two analyses and get, a bigger bang for your buck. So we're working with all four of those traits, and we're making crosses this summer to hopefully funnel those into a genetic background with high yield potential and good agronomic characteristics, because many of the accessions that we worked with in this study have problems with shattering and lodging and disease and other things that make them agronomically undesirable.
It's nice. So a follow-up question. When I look at your, you started off with extremes from a subset of genotypes, and you use those sort of as your goalposts to look at this technology. I really like the variance component approach that you're showing as a way to evaluate, how good the seed is versus the leaf tissue. But how relevant is that range going to be in a breeding program, on one hand, and in maybe, is it possible that there might be even more diversity out there in soybean to further expand that range, where you'd really impact those variance components?
We really haven't looked much beyond the 373 genotypes that we had in our original panel. It's difficult to say. I think that what we have in our panel is very representative of the diversity that's probably in the soybean germplasm as a whole. We know that there are some genotypes that have been characterized in the past as being drought tolerant or very slow wilting. We've looked at their C13 ratio, and they fall more or less in the middle of the panel that we've used. So we feel like we're capturing extremes on both ends.
Well, it sounds good. I'm not seeing any other questions, so we'll give everyone one more chance. If they've got anything, you can go ahead and unmute your microphone, or go ahead and chat to either Larry Purcell or myself. All right. Well, hearing none, thank you, Dr. Purcell. Really appreciate your seminar, and look forward to seeing what else is coming from your lab and your colleagues.
Thank you, Dr. Murray. It's been a pleasure.
Okay. Have a good day. Bye.